So we just discussed regarding two mutations that could be helpful, and I am continuing to discuss with you or teach the chapter on microbial technology. So I almost covered 3/4 of this of the chapter last Tuesday, but I will just do a quick review. So this is these are the this is a list which contains proteins that has been approved to be used in humans. So these are recombinant proteins, right? And you see a lot of interferons and interleukins, which are immunomodulators. Here you see one of the strategies. We have discussed various kinds of strategy using plasmid, using directly cloning of DNA, genomic DNA or cDNA, and then doing protein escalation of protein expression so that you get enough proteins in the system so that it can be used for therapeutic or diagnostic purposes. That's why we are doing recombinant protein, because these proteins are produced inside our inside our cells in a very minute amount, and extraction of protein from, let's say, a cadaver or a dead body is unethical. Same is the case with pituitary. Pituitary is a very small gland which plays a very critical role in secretion of hormones, endocrine hormones, growth factors, and it's difficult to. There's no other way to get those hormones if needed for therapeutic purpose without the help of recombinant DNA technology. So microbes, there can be two kinds of microbes. One are bad microbes, one other is good microbes. Good microbes are sitting in our intestine and various parts of our body that helps in digestion of the food. And food digestion is very critical now because there is a direct correlation between feeling, how we feel every day, how we think, and what are our actions, right? So type of food we eat, the ability to digest food can affect our psychological, neuropsychological, immunological framework, right? So that is very important now. The axis between brain and food, the axis between brain and gut, and if we have good bacteria that digests food, then it's better than there are bacteria who are not efficient to digest food on timely basis. Then besides the bad bacteria, so one thing about bad bacteria is, yes, it is critical that they can cause disease, direct disease, whether it is pneumonia or cholera or tetanus or viruses, fungus. But a good thing about scientific technology is that you can tame these bacteria in the lab and try to understand its biology, and also use its product as a tool. For example, we use reverse transcriptase from HIV that is used to make, this enzyme can be utilized to make cDNA from mRNA. Antibiotics, right? Antibiotics are essentially the metabolites, primary or secondary metabolites of bacteria. So restriction enzymes, the plasmids for cloning, for expression. So good thing about this that scientists can work on these pathogens, bugs, bacteria, viruses, and they can use for developing new medicines or try to understand the biology to develop future medicines. One of the microbe is yeast, which is unicellular eukaryotes, unicellular or multicellular, and some companies, biotech companies, they want to work on yeast model because there are certain proteins which cannot be folded. I remember when I was talking about proteins, protein needs to be folded, right? If they're not folded, so sometimes eukaryotic protein may not be folded nicely inside the bacteria cytoplasm because of different of machinery system. Human cellular machinery requiring protein folding could be very different from bacterial. So yeast is eukaryote, and it can be used to make proteins which undergo post-translational modifications like acetylation, methylation. Bacteria may not have, right? So some scientists, they try to work on yeast system, and for to scale up production of proteins or hormones in the yeast system, we need to do the fermentation. So it's a big mm, um, big chamber which is filled with nutrient, controlled pressure, temperature, and then the yeast, they grow and they try to and you can see the yeast is part of major dairy products, right? Then we started getting into vaccines, right? Some vaccines are very successful. We don't. So there are many pathogens, but we don't have vaccines to every one of them. Right? The reason we don't have vaccine to each one of them is because their life cycle is very tricky. They can trick human immune system. They can trick human transcription system, translation system, and they can use this transcription system or translation system for their own good. So it's until you get a target molecule against which the the therapy could be designed. The research has to continue. But on the other side, there are a lot of positive stories, like polio has disappeared, diphtheria has disappeared, very less occurrence of mumps and rubella. But if you go to tropical countries, every time when season changes, when climate changes, there's uh, so many types of disease that spreads, right? So so these are the list of potential vaccine candidates that people are scientists are still working, right? So vaccines, the major role of vaccines are to create immunological memory. To within our body, they want to create a depot. In that depot, there are cells which are waiting for the real organism to come and attack human body. Once it sees that, yes, they have encountered, then the immunological memory is ready to take on the bug or the virus. Now, these immunological memory cells can also be tricked because viruses can mutate, bacteria can mutate, there can be drug-resistant bacteria. So, yes, vaccine is hopeful. It's a good strategy, but we have to keep on top of pathogenesis of these viruses and bacteria. As the virus is changing, as the virus is mutating, we have to understand its biology. For example, the alpha COVID or delta COVID, and then the Omicron. So we have to understand this emerging pathogens as they emerge. So the basic logic for vaccine is to create memory cells, right? And these memory cells can be living in some lymphoid organs, and when a pathogen goes through blood route, then in the lymph node, these cells can take on the pathogen. So now vaccines, there can be, everything takes its own time, and there was various approaches for a vaccine development. One of the earlier approach was to make the pathogen pathogen should live, but it should lose its pathogenicity, meaning that, yes, this is E. coli, but this E. coli cannot cause disease. So it's a pathogen, but it has lost its pathogenicity. So these bugs, these pathogens are called as attenuated. So although this cannot cause disease, but they can be used as a modality for vaccine development. And these pathogens, which are a foreign body, they're called as antigens, right? A pollen could be an antigen that can cause sneezing or running nose or running eyes, red eyes, itchy eyes, red skin. So these are any foreign uh, particle that can elicit immune response. But there are foreign particles that can trick immune systems. So I don't want to go into immunology. So these attenuated pathogens can work as a vaccine candidate, and they can be mixed with adjuvant. What is adjuvant? Adjuvant is given together with the vaccine. This stabilizes the vaccine inside human body. It is possible that when vaccine by itself is injected, it can be degraded. So this adjuvant, it works for the slow release of these molecules, such that the immunological development of immunological memory is not short term but long term. For example, I can give an example. Let's say if you go to a bus terminal, Port Authority, right? If you have gone, or if you go to a train station, trains, if there is a yard where there is 100 of trains or trains standing there, subway trains, they don't all start working together one by one, so that there is a support system for people to move from one place to another. Likewise, adjuvant, it helps in the slow release of vaccines so that the immunological memory is solid. But if it is not solid, then we have to give booster. Booster, there can be one booster, two boosters, or depends how quickly the pathogen mutates. So one-to-one for the development of vaccine is the pathogen and the human immune system, how they behave with each other. But so that's on the surface, superficial, pathogen and immune cells. But still things are regulated at the level of genes and transcription and translation. So if you go protein, if the strategy is to develop a vaccine to be protein, then it will take time. If the strategy is to take mRNA, so it's in front of us. The vaccine, the epidemic started in March 2020, and now this is we started to take our first dose of vaccine. Maybe healthcare workers, they got as early as December of 2020. Some people got, by 2021, majority of people got. Now, once the vaccine has gone through its development, first, any vaccine is developed in a lab, right? And in the lab, the data has to be very good. It has to be reproducible, and then it goes to preclinical development, like given to mice to check whether the vaccine is protective or not. Then it goes to human trials, phase 1, phase 2, phase 3, and then it goes to manufacturing plant. Now, once vaccine has been made, it has to be made available for two things all around the world, and also it should have more shelf life, right? Because if a vaccine is good for 1 week, look at the cost and effort, and how will it travel from let's say US, New Jersey, or let's say wherever the Pfizer plant is, how will it travel to a small village in India? It will, 1 week is gone. So you have to increase the shelf life of these vaccines. You have to make sure that these vaccines, things fluctuate, temperature fluctuates, many things fluctuate. So these vaccines should stand the test of time with respect to quality control QC. Should be stable. And you can see here, in addition to mRNA, right? In addition to mRNA, there are many things in the vaccine that we received, right? So few of us got Moderna, few of us got Pfizer. So let's say I took Pfizer. So of course we had mRNA coding for the spike protein. Then there were lipid molecules to increase stability, cholesterol, phosphocholine. Then this was most interesting, this very simple formulation buffer. So when you get the vaccine, it is in a solution shape like watery. That water is PBS, potassium chloride, potassium phosphate, sodium chloride, and sucrose. Sucrose is sugar. So maybe people who have diabetes and all those things, so they should wonder that this vaccine is coming, but it could be very tiny amount. In contrast to Pfizer, Moderna has a very different kind of chemicals. It has acetic acid, sodium acetate, sucrose. Sucrose is a good preservative. Sugar is a very good preservative. If we put sugar, then it doesn't let things grow. But again, if it's not stored at proper temperature, even in sugar, fungus or bacteria can grow. So mRNA for both is almost the same against spike proteins, but their ingredients are very different from each other. And then there are various stages of vaccines that can be used. So we talked about recombinant vaccine for hepatitis B virus. Then we talked about subunit vaccines. Once you figure out that envelope proteins are important, then from protein you go at the stage of peptide, right? Because some proteins could be very big and the whole protein may not be immunogenic. What do I mean here is that let's say this is a protein, and by truncating this protein, by making this protein, let me see, I'm just trying to, so let's say we want to see how this, is this whole protein immunogenic or there are a portion of this? So we break this protein into several parts, A, B, C. So you can break this using restriction enzymes, A, B, C, D, E, F. And then through research, you find out that only the fragment uh, uh, only the fragment D is immunogenic. So you want to make vaccine against this. What's the point of making vaccine against these, right? So you'll focus on this because this is generating immune response. So from peptide, you go to from polypeptide, from the protein, you go to small peptide. So this is where the subunit vaccine comes into play. Then you check, through protection assays, whether the vaccine is protected or not. So what you do, you take mice model. You can take monkey model, but monkey model is advanced. Initially, you want to do it in mice model because you can use large number of mice and you can give different conditions. So basically, you take two groups of mice. One, you inject the vaccine. In other, you don't inject the vaccine, but you give viruses to both. You see that the mouse which got the vaccine candidate is protected. So these are the initial lab experiments. These are preclinical experiments. Before going into human body, this has to be determined. So so mice, then possibly rabbit, then possibly monkeys, and then higher mammals, and then you go to human body. So here we talked about GD protein from herpes simplex virus. We don't need the whole protein, but we need only section of protein that can be useful for generating vaccines. So we know that there are 5 domains. So we want to know which domain is more immunogenic. So a small domain could be very less immunogenic. So it can be tagged to a carrier protein called as keyhole limpet hemocyanin. Then we also talked about the complexity with malaria parasites. So with this, we close the section on vaccines and microbes, viruses, bacteria, parasite. Now we go into vitamin C, how microbes helps to make vitamin C or vitamins. So vitamins are not really synthesized in our body in a true form, but we depend upon bacteria. And one of the salient example is vitamin C. And vitamin C is very important for skin. If we are deficient in vitamin C, it leads to a skin disorder called as scurvy, right? So of course, when you go to a physician and he or she recognizes that that patient is suffering from scurvy, then vitamin C will be prescribed. But the question is that why vitamin C is not being made in our body? The answer to this is vitamin C is manufactured by 2 bacteria. To synthesize vitamin C, 2 bacteria are involved. But because of molecular biology and molecular biotechnology, you can make these through gene exchange or gene recombination or transgenic bacteria. You can now make vitamin C through using only 1 bacteria. So I repeat, initially 2 different groups of bacteria, 2 species of bacteria were needed to synthesize vitamin C. But because of molecular biology techniques and molecular biotechnology, a recombinant or a transgenic bacteria was made that could single-handedly generate vitamin C. So 1 bacteria had the capacity to catalyze D-glucose, but this bacteria did not have the capacity to catalyze the last step, that is 2-keto L-gluconic acid to ascorbic acid. This last step could not be done by the bacteria called as Arvenia. So for the last step, you needed another bacteria called as Corynebacteria or Brevibacterium or Arthobacter that had the enzyme 2,5-DKG reductase that would convert 2-KLG to ascorbic acid. So what happened that this particular gene 2,5-DKG reductase was taken out from Corynebacteria. Then a recombinant E. coli was done, engineered genetically, and then that particular gene from E. coli was transformed again into Arvenia. So this Arvenia herbicula now has all the genes which is required in the pathway to generate vitamin C. So not only vaccines, but vitamins also. So as I was mentioning to you that there are bacteria in various parts of our body. There are bacteria, fungus in our teeth that helps in the digestion of milk. There are bacteria in our stomach, large intestine, small intestine that facilitates. So these are called as microbiomes, right? So it is very important that we have a healthy bacteria in our intestine or gut so that the digestion of our food is very timely. If not, then it can affect the feeling, the thinking, the daily life. One can become sluggish, bloated, and may not be very good at getting things done. it affects the psychology, mood, many things. So probiotics are probiotics means one that supports life, right? So so there are gut bacteria that are inside our intestine that helps in digestion, and there are 300 to 500 kinds of bacteria containing 2 million genes which are present inside our intestine, right? And they are totally different field of biology called as microbiome, right? So probiotics is very important. Probiotics, it helps the bacteria to grow, to sustain. There could be small amount of bacteria that helps the food digestion. And some of these probiotics could be, yogurt, well known, then sour cream, cheese. They all can help the bacteria to grow. Chocolate. As I mentioned to you that microbes are present in many organs of our body: oral, skin, digestive. Um, they're present, and particularly in the gut, right? It modulates the nervous system. Digestion is directly converted to nervous system, right? And then it helps in the formation of blood vessels in the intestine, because if the blood vessels are there, then the direct signal bacteria will be healthy, because through these blood vessels, oxygen will reach, and carbon dioxide will go from intestine to outside. So, but if there is no blood vessels in the intestine, then intestine will decay. Right? So these gut bacteria also supports our immune system. So actually, these gut bacteria, it controls our nervous system, immune system, and also psychological system. So it's very important these days. Then microbes are also important for biofuel, right? Because we know that we are dependent upon fossil oil, and these fossil oil can create pollution, increased carbon dioxide that can elevate the temperature of the planet or the neighborhood, and then there will be so we need an approach where there's so much of biomass around us that can be used to develop fuel rather than depending upon fossil fuel. Solar energy, you see, it's getting popular, and people are making sense out of it. So for example, let's say you must have heard that many years back, maybe 10 years back, I don't remember the date, there was a spill of there was a British oil company, BP Oil, in the Gulf of New Orleans, and they had a major accident, and whole sea was full with the crude oil, and a lot of sea organism died due to that, and the whole sea beach and everything was destroyed, or they suffered. So scientists quickly developed engineered bacteria that could eat up those because these layers, these crude oil can sit on the surface of the water for years or months, and they can cut off the oxygen supply. So all the plants which are growing in the bottom of the sea, or those small fishes or aquatic organisms, can die, and also it spreads a lot of toxins in the aquatic climate. So these bacteria can degrade those, digest those products which can be toxic for the environment. Now, lastly, I want to come to this paper, right? So just wanted to give you a brief way to how to read a paper. It's very important to, when you're reading a paper, you read this title, multiple times. Sometimes these titles are like very condensed. Like sometimes I get confused when you see this breaking news on CNN and you want to make, there's short sentences without proper verb and you difficult to understand. So you have to read those breaking news again and again, that what's really is going on. Uh, so especially if you're reading the newspaper, not watching the TV. TV you're listening and reading, but here, so yeah, so these titles you have to, so you may not understand and one time, so you have to read multiple times. So this is urban park soil microbes, a rich reservoir of natural product, biosynthetic diversity. It's pretty straightforward. Urban park soil, so it could be New York Park soil. Are they having bacteria? And does these bacteria have produced natural products which have biosynthetic, which has biosynthetic diversity, right? So, so that's the title. And when you read the title, you should be very curious to know who is the first author, who is the last author. First author is the person who does the work in majority, and senior author is the principal scientist. And you should also see where this study has been conducted. So this was conducted in mainly in New York, but it was led by Rockefeller University, which is on 68th Street and York Avenue. So who is the first author? Zachary Sherlock Powers, and last author is Sean Brady, right? So then again, abstract is the summary. It could be bulleted points in very condensed, but I chose this paper because you can read. And when I read, I try to try to underline those words which could be important, right? Or you don't know, it's not part of your vocabulary, so you need to increase, right? So, so you can read this. So what are natural products? So natural products are those products which are available in nature, but they have activity. Activity means bioactivity. It is active inside the cells. It could be used as a vitamin. It could be used as a medicine. It has a function inside the cells. Then what is non-ribosomal peptides? So there are two kinds of proteins. Number one protein is proteins that are produced by ribosomes. And there are proteins which are not produced by ribosomes. Uh, and these proteins have amino acid which are slightly modified, like formulated. These proteins could be formulated or chemically modified. Polycatides could be secondary metabolites. So this particular scientist looked at the soil in the New York City. And, so read this, you just have to read briefly. Um, what, um, what did he do? He introduced the topic. And here is the significance. What is the importance of his work? Right? So it's just one small paragraph. And so in the end, he summarizes his result, and he says that in the New York Park system, he found antibiotics such as erythromycin, rapamycin, ascomycin, netamycin, and also some anti-cancer and fungal molecule. So how did they come into soil? They came into the soil because they're present because bacteria are releasing these products in our soil. So what is the need for this study? The need for this study is because uh, all the medicines that we are using, uh, there are two things happening. These medicines could be not effective. And secondly, there can be new diseases, right? Cancer is a heterogeneous disease. It cannot, one single bullet cannot kill all the cancer cells. So you need multiple medicine approach to cure cancers. Similarly, antibiotics. We have only few antibiotics which we take. After 10 years, people can get resistant. So based upon these this paper, there are a few questions in your assignment, very straightforward, very simple. Okay. So with this, I finish this chapter. Any questions? And just to remind you that office hours are from 4 to 4:55 on both Tuesdays and Thursdays. And the class starts from 4:55 to 6:05 or 6:10. Anything that you want me to review again? You have any questions? Anything I rushed? It could happen when you're talking extempore. Sometimes you're not able to control the speed. So if you want me to go over something again, I will be happy to go do that. So if there are no questions, may I end this class? Because we are recording and I don't want this to be blank. So I can come back again. I can just end the class and I can come back again because the class is until 6:10. So we still have 15, 20 minutes to go.
Yes, Professor. Yes, Professor, we can end this class.
Okay. I see something
Professor,
in the chat.
Professor, did you have me down Shamika Lawrence?
Let me check. Yeah. Okay.
Okay, thank you.
Welcome.
Professor, did you get my attendance?
May I know your name?
Naful Bajwa.
Yes, I got it now.
Thank you.
Hi, Professor Ruhama Sufran.
Okay, how are you?
I'm okay, thank you. How are you?
Good.
Okay. Have a good evening.
You too, Rahama.
Thank you.
Bye.
Professor, did you get me on attendance?
May I know your name?
Yael Nakhshumo.
Yes, I got it.
Thank you, Professor. Have a good day.
You too.